Hello everyone, myself Dr. Makhan Singh, Associate Professor Botany, Government College, Mahendragarh. Today in this video lecture we will discuss about the mutations. First of all, we see some pictures or deformities we generally see in the nature. This one is the picture of strawberry, where we can say a multiple fruits are aggregated in a single structure. In this structure of Decascarota, there are branching. The routine, there is a single conical form of the root, but here it is branched structures. This is also due to the mutation. In this cone, the structure which should be a single structure, but here the two structures are present. That is also a result of mutation. Here in a very fantastic structure of apple, that it is two colored. Half one is the light green color, another is the routine red color structure. These modifications in these fruits and these structures is due to a certain change in the genes or certain chromosomes. In this flower, the two colors, half is white and half is pink, means the change in the color is due to certain change in the genes. Here in this plant of cannabis, the two colors are present in the same leaf. This is the leaflets, and the two leaflets are yellow colored and other three are green colored. In this leaf 1, 2 and 3, these three leaves are yellow colored, and the fourth one is half green and half yellow colored. Means there are certain same genes which are controlling the change in the color. Here in this plant, the different color pigmentation into the leaves. The color of the tulip with this petal contains two colors, yellow and red. This is also due to the change in genes. Now we can see the example of animals only. Animals also. This snake contains two heads. Means there are certain changes, deformities in a certain chromosome or gene which results in such type of deformities or external appearance of the characters. This is the polydactyly case, which is also the change in the genetic constituents which results in the deformity. These three fingers, these are fused, and these two fused. Here these two fused means polydactyly. The number of fingers is more than a common routine. It means certain genes or chromosomes, they are altered or they are stimulated to change or behave abnormally, and during the embryonic development these resulted into the polydactyly case. Generally we think these changes are common, so we conclude these points by this. As the scientists learn to read the instructions in our genes, they are discovering that much of our DNA is riddled with errors. It means the DNA present in our genome that contains a large number of errors, means the mistakes, means which are not common or routine. We each inherit hundreds of the genetic mutations from our parents, as they did from their forebearers. Means the genetic combination which are we receiving from the parents already have mutations, and those mutations which we received from their forebearers, they are coming to us, and certain other changes. In addition to this, the DNA in our own cells undergo an estimated average 30 new mutations during our lifetimes. This is very important thing, that the mutations, they are continuing not only in our forebearers and our parents and grandparents, but we are also facing the same situation. Means a general trend is that an average 30 new mutations during our lifetime we are also undergoing this process, either through the mistakes during the DNA copy or cell division, or more often because of the damage from the environment. These are the simple factors which results into these 30 mutations. When are the mutations harmful? During the copying, bits of DNA may be deleted, inserted, broken or substituted. These are some common processes which happens in a routine exercise, that when the DNA is replicating, then some DNA may be deleted, inserted, broken, or it may be substituted by some certain other DNA segment. Most of the mutations affect only the parts of DNA that do not contain instructions for making genes. Means generally we see that the DNA segment present totally on the chromosome is not coding for the genes, it is coding for the proteins. Those non-coding area which go under mutations, that doesn't affect the organism. But when the coding area where the DNA segment is coding for particular gene, then it becomes a problem. So the DNA which is non-coding area, which is going under mutation, so we need not to worry about them. Problem arises only when the DNA enters a message that tells certain cell to manufacture a certain protein. Means the coding areas from DNA to RNA and RNA to proteins are synthesized. Then if there is a change in that coding area or that particular segment of DNA, which will result into the altered mRNA, and altered mRNA will construct a particular type of protein, that may be a problem. Such messages are spelled out in varying sequences of 4 chemical bases that make up of DNA. Means those nitrogenous bases, that is adenine, thymine, guanine and cytosine, that is purines and pyrimidines, they changes the message when the DNA is altered. The mutations occurs as random chance event and cause changes in the DNA sequences. If the mutation occur in gametes, they can be passed on to the offsprings, means from that generation to the next generation. Mutations may be caused by exposure to toxins or radiations. Mutations may be in any form. They may be the toxins, chemicals, they may be biological, that may be physical, that may be chemical, that may be in the form of radiations, which is responsible for change in the DNA constituents. Some mutations causes disease, yet other may lead to the variation and organisms that make it better adapted for their environment. Mutations are necessary also. Certain change in the genetic constituency, means GM crops, GMO, which are called as genetically modified organisms, they are also useful. High yielding varieties, they are as the resultant of the mutations. But some mutations, they may cause disease. You see in this photograph that these are some deformities due to the change in the as a result of mutations. Now we discuss about the kinds of mutations. In a broader sense, there are 2 kinds of mutations. One is the chromosomal mutation and another is the gene mutations. The chromosomal mutations, which occurs into the chromosomes, their number, and gene mutations means in the constituents of the genes. So now we discuss first of all about the chromosomal mutations. The chromosomal mutations, they are the abnormal change in the structure of all or part of the chromosome or in the number of chromosomes of an organism has. Means the number may be changed or its structure may be changed. It may be deleted, it may be inserted, it may be duplicated, it may be translocated, but the number may also be, means some certain chromosomes may be lost, certain new chromosomes may be added, means that all type of the mutation is called as a chromosomal mutation. For example, as we see in case of human, there are 23 pairs of chromosomes, where 22 are the pairs are the autosomes and 1 pair of the sex chromosome. Human with the Down syndrome have 47 chromosomes. Means the Down syndrome has a trisomy that is called as 2n plus 1. Means here the trisomy of 21st number of autosome. These are the karyotyping structures where the chromosomes have been differentiated into different groups. So we see in this group which is G, where the 21st number of chromosome, there are instead of pair, one extra chromosome is added. This 21st chromosome is as the resultant of trisomy. So the total number of chromosome is 2n plus 1 is equal to 47. And what will it result in? It results into the Down syndrome. The disease or the deformity is called as the Down syndrome due to the trisomy of 21st chromosome. What happens in this trisomy? Extra 21 chromosome. One chromosome is added, and the rate of this Down syndrome is, it affects 1 out of 700. Means among 700s it is 1. Alters child's phenotype. If the genotype has been changed, means a 1 chromosome has been added, so 47 chromosomes are there, so it will result into the phenotype, changed phenotype, and the characteristic facial features, short stature, size will be the short, usually some degree of mental retardations. They are also observed in this Down syndrome. Now we discuss in brief about the gene mutations. Gene mutations mean only the gene present on the chromosome, they have some changes. So the gene mutations, a change that affect a gene on a chromosome. It causes the cell to produce abnormal proteins. Means when the gene is changed, it means the coding area of that particular gene that will also produce a abnormal protein, which is either not necessary or maybe harmful for the organism. There are two types of gene mutations. One is point mutation and another is frame shift mutation. So first of all, we discuss about the point mutation. In the point mutation, a gene mutation involves only a single nucleotide. For example, ACA mutates to read ACT. In this case, ACA is coding for a particular amino acid, and here we have changed this adenine replaced with the thymine. Means this purine is replaced by the pyrimidine. mRNA codes for ACT. This is the sense strand, and this is the codon, and anticodon on mRNA. It will be the UGA, and UGA codes for a stop codon. It means the ACT, when this particular amino base triplet, it is mutated with only a single gene, single nitrogenous base, then it has been completely changed. That it is stopping the synthesis of proteins, means the chain is shortened. This will cause the necessary proteins not to be made. Means further the chain is stopped, it means further protein synthesis and the production of the enzymes that is stopped, and the growth or progress of the organism will be halted. Here in case we see that this base triplet UAA, this is coding for leucine, proline, CCA, ACC and so on. These are the base triplets. single base deletion, here this C is deleted. This C, which is in orange color, this is deleted. Then how will be the reading? This C is replaced, then the C further comes with the A and CAG, means the change has been started from here, and the resultant here, the protein chain, nucleotide chain is so long, but here after the deletion of only one gene, the chain is shortened, because this UGA, this codon, this is for codes for nothing, and it is a stop codon. The chain is terminated here. So the single chain point mutation that has been shortened the protein chain. Point mutations, or it is divided into two parts. Point mutations, it may occur in the non-coding region or in the coding region. If it occurs into the non-coding region, just a mutation, simple mutation, no change in the protein and no functional consequences. There are no observing, means the phenotypes are not observed in if the mutation is happening into the non-coding region. Point mutations can take place at a different localities in the genome, coding and non-coding regions. If the point mutations, they are occurring in the coding region, then what will happen? Non-synonymous mutations, they are called as non-synonymous mutations. That may be missense mutation or it may be nonsense mutation. What are the missense mutations? Means change an amino acid into another amino acid. Means the sense strand that is producing the another amino acid in place of previous one. That is a change. And in the nonsense mutation, change an amino acid into a stop codon. When the mutation occurs into the nonsense, means when the change into the nucleotides or the nitrogenous bases, that results in such a way that the resultant is the terminator codon. Chain is terminated. Here the chain is stopped. Here one amino acid is replaced by another or changed into the another. Here we see that how the substitution means one nitrogenous base is replaced by another, means is substituted. The total number is same, but it is substituted. Here the nitrogenous base adenine and guanine, these are the purines, and cytosine and thymine, these are called as the pyrimidines. If the adenine is replaced or substituted with guanine, then this is called as transition. If cytosine is replaced by thymine, means purine is replaced by purine, or pyrimidine is replaced by pyrimidine, that is called as transitions. Is that adenine replaced by guanine or vice versa, guanine replaced by adenine, means purine replaced by purine, that is called as transition, and cytosine replaced by thymine or thymine replaced by cytosine, means the substitution of pyrimidines only, that is called as transition. But when adenine is replaced by cytosine, means purine is replaced by pyrimidine, then it is called as transversal. Purine replaced by purines, that is transition. Pyrimidines replaced by pyrimidines, that is also transition. And purines replaced by pyrimidines, that is called as transversion. Or pyrimidines replaced by purines, they are also transversion. Here the adenine replaced by cytosine, this is transversion. Adenine replaced by thymine, that is also transversion. Guanine replaced by cytosine, transversion. Guanine replaced by thymine, transversion, or vice versa. Thymine replaced by guanine, also transversion. Thymine replaced by adenine, transversion. And cytosine replaced by guanine, transversion. And cytosine replaced by adenine, also the example of transversion. So here we will discuss some queries. what is the change from adenine to guanine? Here we see that adenine is a purine and guanine is also purine. So the change from purine to purine or pyrimidine to pyrimidine, it is called as transition. What is the change from adenine to cytosine? Here the cytosine is pyrimidine, adenine is purine. So change from purine to pyrimidine, it is called as transversal. It is called as transversal. Here we see that adenine is replacing the cytosine, then it is transversal. What is the change from adenine to thymine? Means this is purine, this is pyrimidine. Again it is the case of transversal. What is the change from adenine to adenine? Means purine is changing purine itself, then it is the transition. Now we discuss about the second type, that is the frame shift mutation. The frame shift mutation, it is caused by the addition or deletion of nitrogenous bases in DNA or mRNA. The shift of the reading of the codons from the site, here the color has been changed. This is showing that the change is started from the site of change onward. Means the codon from at the site, at the point where the codon has been changed, then the shift, total shifting of the onward codon is changed. This is called as frame shift mutation. This may also be due to the insertion or deletion of one or more nucleotides, not only the nitrogenous bases, but due to the one or more nucleotide has been changed, then this will also result into the frame shift mutation. For example, the cat ate, this is one single sentence, the cat ate, in a frame shift mutation it would read as the, here this C has been deleted, this C has been deleted, then what will be the reading? The change has been started from here, then the base triplet, next base triplet is ATA and T is free, which is a meaningless, means no meaning of this line. This line contains a meaning. Which letter was deleted? The letter was C. Further, here we see the cases of frame shift. Look here, there is a sentence, the cat ate the rat. This is a sentence which is a meaningful sentence, the cat ate the rat. Here we are changing a little change, the cat ate the rat. The meaning of the sentence is same, as we are pronouncing that the C has been replaced by K, but the resultant is, the meaning of the sentence is same, but one change is here, and this is not resulting into any phenotypic change or any meaning of the sentence. This type of mutation is called as silent. The cat ate the rat, similar sentence, meaningful sentence, but when this C is replaced by H, the hat ate the rat, means this is the case of substitution. The sense of the sentence is changed, means the hat ate the cat and then the hat ate the cat. This is the substitution. Here the cat ate the rat, the cat ate the rat, and the CAA tat her at, it means this T has been deleted. So what will be the result? The CA same, this is the change started from here, CA, T is deleted, then this A is coming in this base triplet, then the T containing the next T, and then this 2 containing the next word, and the resultant is, the sentence makes no sense. The CA tat or rat, what does it mean? It means the shift from the change onward has been changed. The cat ate the rat, and here one E is added, extra added, inserted, then what is the reading? The cat at the rate, the ECA, TAT, ETH, ERAT. What does it mean? No sense. Hence, the reading from the change onward has been changed. This is the frame shift mutation. The silent mutations and substitutions are point mutations, and insertion and deletion are frame shift mutations. These two cases are the frame shift mutation, and these two cases are of the point mutation. In case of silent, there are no change, it will remain the same, and in case of substitution, the sense of the sentence has been changed. Here in this case, this is case of non-muted beak in this bird, and here the beak has been mutated and the bird is capable to feed on the prey. So this was all about the mutations a little. Further we will discuss about the mutations and its applications, causes into the next video lecture. Thanks.